The last big concept from chapter 15 is the fact that genes that are physically located on the same chromosome tend to be inherited together. We call these linked genes. So in my picture here, the recessive A and recessive B are linked together. When doing experiments with fruit flies, the Morgan group noticed that body color and wing size traits are usually inherited together. So if a parent had both a gray body and large wings, those two traits passed as a unit to their offspring. In other words, these genes did not seem to assort independently. Now, the basis for this is that those genes are actually on the same chromosome. What could happen to these chromosomes that could separate those traits from one another? Well, it could be crossing over. If crossing over happens somewhere in between those traits, then we could get the recessive allele from mom and the dominant allele from dad together in one recombinant chromosome. The Morgan group was able to do experiments to test whether different traits were inherited together as a unit or if they assorted independently. So to set up this cross, they started with parents that were true breeding for two different traits. So these guys are homozygous dominant for body color and wing trait, and the other parent is homozygous recessive for body color and wing trait. So a little confusing thing about fly nomenclature. The gene for body color is named after the mutant. So the mutant phenotype is black. So the gene that controls body color is the B gene, or the black gene. And a plus indicates that that's the wild type. So the ones that are gray are B+, and the ones that are black are just B by itself. So homozygous recessive B is black body type, and homozygous dominant B+, is the dominant phenotype, true breeding parents. So they set up this cross with parents that were homozygous dominant for both body color and wing trait. VG is vestigial, meaning little wings that don't do anything. So the mutants that were homozygous recessive for the VG gene had vestigial wings. The flies that were homozygous dominant had normal functional wings. So when they set up the cross, they got an F1 generation that was heterozygous at both the B locus and the VG locus, and had the dominant phenotype for both body color and wing trait. And then they set up a test cross to determine whether body color and wing trait were inherited together as like one gene, as one unit, or if they were controlled by separate outcomes, separate chromosomes, separate genes, separate traits. So there's two possibilities that could happen here. It's true that body color and wing trait were inherited as one piece, or if one gene controlled both of these traits at the same time, then whatever versions came from the P generation up here would be stuck together down here in the F1s. So when we did the test cross, the dominant body color trait and the dominant body color trait and the dominant wing trait would be stuck together. And then from this parent in the test cross, you get recessive and recessive. So these offspring would be heterozygous at B, heterozygous at VG, and have the dominant phenotype for both. And then if little b and little VG were stuck together from this parent, the other possible outcome would be that you get recessive and recessive from this parent and recessive from this parent, and these offspring would be recessive for both traits. So if the traits were tightly linked, those were the only outcomes we could see. But if the genes assort independently, then B+ could go with VG+, or B+ could go with VG. So you could have B+ coming from this parent, little b coming from this parent, and V+ coming from this parent, and little v coming from this parent. These offspring are heterozygous. You could get little b, and little VG going into these guys being homozygous recessive. But you could also get B+ from here and little b from here, heterozygous at B, with the recessive allele for wing trait and the recessive allele from wing trait over here. So these guys would have dominant body color, but recessive wing trait. Or you could get the little b from this parent and the dominant VG from this parent, and you could end up with offspring that were recessive for body color but dominant for wing trait. Okay? So these guys over here that have a unique combination of traits that were not seen in the P generation or the F1s are called the recombinant offspring. They have a combination of traits: dominant body color but recessive wing trait, or recessive body color and dominant wing trait. So these are the two expected outcomes, but what the Morgan group actually got was, for the most part, it looked like the genes were linked. Usually they got either all dominant or all recessive. But occasionally, and not insignificantly, they got a good chunk of offspring that were recombinant phenotype. So what could account for the fact that usually they acted like they were linked, but sometimes they didn't? The body color and the wing trait genes are both on the same chromosome. So most of the time, those heterozygotes would give either dominant body and dominant wing trait together, or recessive body and recessive wing trait together. So these offspring would be heterozygous at both loci and dominant for both traits, and these offspring would be homozygous recessive for both loci and recessive for both traits. During meiosis, some of the time, crossing over happened in between the B locus and the VG locus. And then you would get sister chromatids that were not identical. They had some of one parent's DNA and some of the other parent's DNA. And when these recombinant chromosomes were used at fertilization during the test cross, you would get offspring that had the dominant trait for B but the recessive trait for VG, or the recessive trait for B and the dominant trait for VG. But this only happens some of the time. If crossing over happened back here, B plus and VG would stay together, and B and VG would stay together, and you would still get the parental phenotype for these two traits. So where these genes are along the length of the chromosome and how far apart they are actually determine how frequently recombination and recombinant phenotypes in the offspring happen. But when the traits stay together in the same way that they were in the parents, we call that the parental phenotype. So for genes that are on the same chromosome that remain linked, we would see the dominant trait for, shorter stay and body color and vestigial and other traits. For genes that are on different chromosomes, on totally different chromosomes, if we think about how likely is it that the dominant A trait is going to stay with dominant D, or is it going to go with recessive D? Okay? Well, it's a 50-50 chance, right? So either dominant will stay with dominant, they'll stay linked, or dominant will go with recessive. So write this down. The highest value you can get for recombination frequency is 50%. It maxes out at 50%. Either it stays with or it goes to the other one. It's a 50-50 chance which one it'll stay with. Okay? So this is true for genes that are on different chromosomes. They will have a recombination frequency of 50%. But for genes that are on opposite tippy ends of the same chromosome, they also have a recombination frequency of 50%, because crossing over happens between each pair of homologous chromosomes during meiosis. So there's a 50% chance that A plus will stay with VG plus, and a 50% chance that A plus will go with VG. Genes are on different chromosomes, they'll assort independently. So you could get dominant Y with dominant R, but you could also get dominant Y with recessive R. You could get recessive Y with recessive R, or you could get recessive Y with dominant R. But when genes are on the same chromosome, they tend to stay together or remain linked. The thing that breaks them apart is crossing over. So if crossing over happens somewhere in between these two linked genes, that's when we'll get our recombinant chromosomes that are half mom, half dad, and have the recombinant phenotype in the offspring. So crossing over is going to separate linked genes from one another. And the closer together two genes are, the less likely it is that crossing over will happen somewhere in between them. The further apart two genes are, the more likely it is that crossing over will happen somewhere in between them. I say that again, write this down. The farther apart two genes are, the more likely it is that crossing over will happen in between them. In other words, they'll act like they assort independently. But the closer together two genes are, the less likely it is that crossing over will happen between them, and the more they'll be linked. So the further apart they are, the higher the likelihood of recombination, the higher the likelihood of crossing over, the higher the likelihood that they'll act like they're independently assorting. But that value maxes out at 50% because A plus is either going to stay with B plus or it's going to go with recessive B. So it maxes out at 50%. Calculate that recombination frequency. You take all of the non-parental phenotypes, so the ones that are dominant for body color but recessive for wing trait, added to the ones that are recessive for body color but dominant for wing trait. So all of the ones that have the the non-parental or the recombinant phenotype. And then you divide the total number of recombinants by the total number of offspring. And that value is what percent of the population didn't look like the parents. Okay, that's the recombination frequency. But this value for recombination frequency maxes out at 50%. You can use the information from recombination frequency to draw a genetic map. Linkage maps can tell us the relative position of genes along the length of the chromosome. They're based on recombination frequencies, but they're not physical distances. So if you try to add them together, they don't exactly add up because there are some recombination hotspots where recombination is more likely to happen that make genes look like they're further apart. And then there are recombination dead spots where crossing over almost never happens, like the centromeres, which make the genes look like they're actually tightly linked and really close together, whereas there may be many nucleotides between them. So in this example here, the recombination frequency between black and cinnabar is 9%, between cinnabar and vestigial is 9.5%. If you add that up, it's 18.5%, but the recombination frequency between black and vestigial is only 17%. So these values don't have to exactly add up because they're not physical distances. So to place genes along the length of the chromosome, you usually have to compare 3 genes to each other. So between black and cinnabar, there's a recombination frequency of about 10%. The recombination frequency between black and vestigial is about 20%. But we know that vestigial is to the right of cinnabar and not over here because there's also a recombination frequency of 10% between these 2 genes. If we looked at the recombination frequency between black and brown, it would max out at 50%, okay? But we know that it's over here because of the recombination frequency between brown and vestigial. So by comparing 3 genes to one another, we can draw a linkage map. So let's do an example of that. Problem, it tells you that these 3 genes are all on the same chromosome. So start by just drawing a line. And then it tells you that the recombination frequency between A and B is 28%. And then it tells you the recombination frequency between A and C is 12%. So where does C go along the length of this line? So C could be right here, or C could be over here. And it's not until we calculate the recombination frequency between C and B that we can determine where C exactly is. If C is in between A and B, then we would expect a recombination frequency between C and B to be around 16%. But if C were all the way over here, we would expect a recombination frequency of 40%. So, given the information in the problem, it's either 16% or 40%. We don't know where it is. Now, if it told you the recombination frequency between C and B is 40%, then we would know for certain that the C locus must be all the way over here. Just a note, it would be equally correct if you wrote B, then A, then C. Okay, so we can flip the chromosome around and that doesn't change where they are on the chromosome. Let's do one more problem. So in this case, A and B have a recombination frequency of 35%. B and C have a recombination of frequency of 35%. What would we expect the recombination frequency between A and C? Possibilities. One is that the C gene is right next to A, and so if that were true, we would expect a recombination frequency of about 0% or very low. But if C were all the way over here, what would we expect that value to be for the recombination frequency? If you said 70%, you're wrong. Why? Because recombination frequency maxes out at 50%. That's the highest value you can get. They can be more than 50 map units apart, but the recombination frequency for any 2 genes, when they're either on opposite ends of the same chromosome or on different chromosomes, maxes out at 50%.